Hello, this is part 5 of Unit 4 UGC-NET GRF Forensic Science. In this part, we will discuss about plant poisons and metallic poisons. Poisons are classified on the basis of their action on the body as corrosives, irritants, and neurotics. Corrosive poisons are further classified into strong acids, for example, sulfuric acid, hydrochloric acid, nitric acid, etc., and strong alkalis, NaOH, sodium hydroxide, potassium hydroxide, etc. Irritants are divided into inorganic and organic. Inorganic irritants can be non-metallic, that are phosphorus, chlorine, bromine, etc., or metallic, that are antimony, arsenic, bismuth, etc. The organic irritants are vegetable poisons such as castor, animal poisons such as snake venom, miscellaneous such as glass powder, diamond dust, etc. Neurotics are further divided into three: cerebral, spinal, and cardiorespiratory. Cerebral ones are narcotics, inebriant, and deliriant. Narcotics are opium and its alkaloids, inebriant such as alcohol, pesticides, deliriant such as datura, cannabis, etc. Spinal poisons are stimulants and depressants. Stimulants such as nux vomica, depressant galcium. Cardiorespiratory can be cardiac or asphyxiants. Cardiac are aconite, oleander, etc. Asphyxiants are carbon monoxide, carbon dioxide, hydrogen sulfide, etc. Some facts based on plant poisons. So, Calotropis produces milky latex sap which causes eye inflammation, keratoconjunctivitis, and reversible vision loss. Calotropis is an irritant. Strychnine is a neurotoxin and it hinders the functioning of glycine and acetylcholine receptors. Yellow oleander contains cardiac glycosides. These are the cardiac glycosides. Atropine is used as an antidote for mushroom poisoning, cholinergic drugs. Datura toxicity in increasing order, it is leaf, roots, fruits, and then seeds. So, seeds of the Datura is the most poisonous part. Datura poisoning resembles atropine poisoning. Sodium and potassium hydroxides are strongly corrosive due to their solvent action on proteins, saponifying action on lipids, and ability to extract water from the tissues. Phenol is corrosive because of its protein degenerating effect, and the confirmatory test for phenol is Weir's nitrate test. William Withering identified digitalis, which is a cardiac glycoside and is originated from herbaceous perennials commonly known as foxgloves. The classification of poisonous plants based on their action is neurotic, that is Papaver somniferum, spinal, Strychnus nux vomica, cerebral, Cannabis sativa, Erythrozolan cocca, Atropa belladonna, and Datura fastuosa. Cardiac poisons are Nicotiana tabacum, Aconitum napellus, Digitalis purpurea. Irritants are Abris precatorius and Calotropis. Miscellaneous, ergot and oleander. On the basis of chemical constituents, plant poisons are classified as alkaloids, glycosides, toxic proteins, and resins. In alkaloids, there are opium alkaloids, phenanthrene and isoquinoline, pyridine alkaloids, uh, which are pyrrolidinotropine, cocaine, datura, atropine. Another alkaloid is indole, which is Strychnus nux vomica, pyridine and piperidine alkaloid Nicotiana tabacum, quinoline cinchona officinalis. Glycosides are digitalin, oleander, calotropis, toxic protein, Abrus precatorius and calotropis, resins, cannabis sativa. Common plant poisons, their active components and their acidic or basic nature. So, Papaver somniferum, its active components are opium and its alkaloids, and it is basic. Nicotiana tabacum, nicotine, basic. Strychnus nux vomica, also known as cuchilla. The active components are strychnine, brucine, and it is basic in nature. Erythroxylin coca, the active component is cocaine and is basic. Atropa belladonna, active component atropine and is basic. Datura fastosa, Datura fruit is also known as thorn apple. The active components are hyoscine or scopolamine, hyoscyamine and atropine. It is basic. Digitalis purpurea, digitalin, acidic. Oleander or canare, thevetin, thevetoxin, cerebrin, acidic. Calotropis, eustarin, calotoxin, calicin, acidic. Abrus precatorius or rati or jacurity bean or rosary pea. The active component is abrin and it is acidic in nature. Cannabis sativa, the active component is tetrahydrocannabinol or THC. It is acidic. Aconitum napellus, active component is aconine and it is basic. Claviceps purpurea, ergot alkaloid, which contains ergotoxin, ergotamine, ergometrin and is basic. Semicarpus anacardium, which is also known as marking nut. It is the active components are semicarpal or bilavonol. It is basic in nature. Chrysanthemum cinerarifolium. The active component is pyrethrin and it is neutral. Thavatia peruviana. It is a cardiac poison. The active component is nerifolin and it is basic. The microscopic examinations of plant poisons. First is morphine. With 5% mercuric chloride, it forms tuft-shaped crystals. With potassium mercuric iodide, needle-shaped crystals. 5% potassium iodide, orange plates. Strychnine with mercuric chloride forms curved ferns. With potassium mercuric iodide, it forms wedge-shaped curve. With gold chloride, it forms feathery rosettes. Nicotiana tabacum with potassium triiodide form nail-shaped crystals. Aconitum napellus with gold chloride forms rectangular prism or golden yellow crystals. With 1 drop of 0.1 normal HCl and 2 drops of 5% sodium carbonate solution, it forms rosette-shaped crystals. Brucine with 2 drops of acetic acid followed by 1 drop of alcohol and 1 drop of methyl iodide forms rosette-shaped crystals. Cocaine with few drops of saturated solution of alum and 1 drop of potassium permanganate forms rectangular pink crystals. Datura with 1 drop of glycerine forms eyelid-shaped crystals. Cannabis with 1 drop of dilute caustic soda results in the formation of cystolytic hair in charas and ganja. Color test or spot test for plant poisons. First is Proddy test, in which the reagent is 50 mg molybdic acid or sodium molybdate in 10 ml of hot concentrated sulfuric acid. With opium alkaloids, violet to green to blue color is observed. With calotropis, deep green to blue to green, and with ergot, it forms deep green to red to grey and then blue. Marquis reagent, formaldehyde and concentrated sulfuric acid in the ratio 1:9. With opium, it gives brownish purple morphine purple, codeine violet, thebaine orange, Arbus pectoris pink, and ergot brown. Mandolin, 1% solution of ammonium vanadate in concentrated sulfuric acid. Strychnine gives deep violet to yellow color, and ergot gives purple brown color. Nitric acid with brucine gives reddish yellow and then yellow color. With morphine and codeine, it gives orange color. Vitalis reagent is 1-2 drops of concentrated nitric acid added to the sample and then evaporated, and alcoholic caustic potash was added. With brucine, datura and atropa belladonna, it gives violet, orange red and then red color. With ergot, it gives dull orange, yellow and purple color. Mayer's test, in which potassium mercury iodide prepared by dissolving 1.357 grams of mercuric chloride and 5 grams of potassium iodide in 100 ml of water, it gives white or yellow PPT with Nicotiana tabacum. Fast blue bee test, 5% ethanolic solution of fast blue bee salt is added, followed by 2 drops of aqueous potassium hydroxide solution. With Abrus precatorius, it gives red which changes to orange color, and with cannabis, it gives purple red color. Huysman test, in which the dried extract of the sample to the dried extract 2 drops of concentrated sulfuric acid heated and then cooled, and then 1 drop of concentrated nitric acid along with potassium nitrate crystal is added. Opium alkaloids give reddish violet which changes to blood red and then to reddish yellow color. Urotropin test, aqueous urotropin solution is used. With opium alkaloids, it gives purple color which changes to blue and then to green. Gerard's test, in which 2% mercuric chloride solution is added to hyoscine, it gives yellow color which changes to red on heating, and with hyoscine, it does not give any reaction. Pallets test, in which 2.5 grams syrupy phosphoric acid and zero point 0.1 gram of sodium molybdate are used as for reagent. Aconitum napellus gives violet color. Alvarez test, in which few drops of bromine are added to the extract and it is evaporated. Then 1 ml of nitric acid is again added and evaporated, followed by 1 ml of saturated alcoholic solution of sodium hydroxide, and again the solution is evaporated to dryness. The residue is cooled and 10% copper sulfate solution is added. In this test, Aconitum napellus gives green color. Van Urk test, in which 1 gram of para-aminobenzaldehyde in 100 ml ethanol and 10 ml of hydrochloric acid are used. Abrus precatorius gives green to blue color. Keller's test, in which 1 drop of concentrated sulfuric or phosphoric acid is added and the solution is warmed on a water bath. Oleander gives pink for nerin and yellowish brown color changes to pink for thevetin. Scott test and Wagner test, 2% cobalt thiocyanate in water and dilute it with 96% glycerine. Sample with 10% of iodine aqueous solution. So for Scott test, the test is with cobalt thiocyanate and water glycerine ratio 1:1. Cocaine gives pink color. For Wagner's test, to the sample 10% iodine aqueous solution is added. Cocaine salt gives brown PPT, and the presence of cocaine is showed by blue PPT. Duquesnoy's Levine test, in which 5 drops of acetaldehyde and zero point 0.4 grams of vanillin in 20 ml 95% ethanol were used as reagent. Cannabis gives violet color in chloroform layer. Other techniques for the identification of plant poisons are TLC, HPTLC, GC, HPLC, MS, HPLC, MS, GCMS, FTIR, and so on. Now, metallic poisons. Lead poisoning is also known as plumbism, saturnism, colica pictorium, Devon colon, painter's colic. Copper poisoning is known as copperitis. Zinc poisoning is known as hyperzincemic. Minamata disease and Mad Hatter disease are associated with mercury poisoning. Itai disease is due to cadmium poisoning. Now the common metal test. First is Reinsch test, in which sample is taken in water. Then 20 ml of concentrated HCl is added along with copper strip at one end of the porcelain basin, and the copper strip is dipped in the sample. The end of the copper strip is dipped in the sample. So if the copper strip turns steel grey, it indicates the presence of arsenic, and octahedral crystals are observed in the microscope. Bluish black deposit on copper strip and needle-shaped crystals in microscope are indicative of antimony. Grey deposit is indicative of bismuth. Silver deposit and microscopic round globules are indicative of mercury. Marsh test to the sample in the porcelain basin or china dish. The 2 to 3 drops of 0.5% pyrogalol solution is added, followed by 1 ml of saturated sulfurated water, and it is boiled for 30 minutes. A current of approx 4.5 ampere at 6 volts was passed for 30 minutes. Mirror of black metallic luster on the other side of Marsh tube is observed in the case of arsenic and antimony, and the respective crystal uh, for arsenic is octahedral and for antimony it is needle shaped, that these crystals are observed under the microscope. Now, metal poisons. First is arsenic. The most commonly found form of arsenic is arsenic trioxide, that is white arsenic, or arsenic sulfide, real gar. Meese lines or transverse white lines are prominent in finger or toenails because of the deposition of arsenic in keratin-rich tissues in case of arsenic poisoning. Arsenic is identified by Gutset test, in which 1 ml of wet digestive sample to the 1 ml of sample, 2 pellets of pure zinc and 5 ml of diluted sulfuric acid is added, and the resultant is passed over lead acetate paper to absorb H2S gas, and it is reacted with mercuric chloride paper, and yellow stain is observed. For antimony, the tests are rhodamine B test, in which to the sample solid sodium nitrite followed by 1 ml of 0.01% rhodamine solution is added. The bright red color of the reagent changes to blue. Another test is phosphomolybdic acid reagent test, in which when reagent is added and heated, the color of the reagent changes to blue. Test for aluminium. Test with aluminon reagent. The reagent is prepared by saturated solution of ammonium salt of orine tricarboxylic acid. A bright red precipitate is observed. Test with alizarin. The reagent is 0.1% aqueous solution of sodium alizarin sulfonate. When few drops of reagent are added to the sample, violet color appears. With excess of acetic acid, the color disappears. And 1 drop of acid is added in excess when a red precipitate or coloration is observed. Test with Morin's reagent. So the reagent is 0.1 gram Morin hydrate dissolved in methanol. To the sample, few drops of reagent are added and green fluorescence is observed. With the addition of 2-3 drops of acetic acid, the intensity of fluorescence increases. So there are 3 main tests for aluminium: aluminon, alizarin and morin. Test for cadmium with hydrogen sulfide. The bright red color of the reagent changes to blue. The reagent is rhodamine B solution. Test with dinitro para diphenyl carbazide. The reagent is 0.1% DPDC solution in ethanol and a brown precipitate is formed which changes to greenish blue. Chromium is tested with diphenyl carbazide and it forms violet or red color. With chromotropic acid, a red coloration is observed. For mercury, the extraction of mercury is done by Fresenium and Babbo method. To the visceral mercury sample, concentrated HCl and solid potassium chloride are added and these are heated in a water bath. KCl3 is added time to time and the resultant is filtered and washed with water. Sodium sulfite is added to reduce the excess of chlorine into HCl. The liquid is warmed on a water bath and air current is passed to expel the excess of SO2. Mercury is tested with diphenyl carbazone to form violet or blue fleck, and with cuprous iodide it forms red or orange color. Tests for copper. First is Piegel's test, in which 2 drops of aqueous solution of the sample is mixed with 2 drops of dilute zinc nitrate solution and 2 drops of mercury ammonium thiocyanate reagent. Pink to violet colored precipitate is observed. With rubinic acid, the aqueous solution is first spotted on a Whatman filter paper number 1 and then the resultant is dried and sprayed with rubinic acid and exposed to ammonia vapor. An olive green colored spot is obtained. Test with cuproin or 2,2-diquinolil. A drop of a test solution. The pH of the sample solution should be above 3 and it is placed on a spot plate with few crystals of hydroxylamine hydrochloride and 3 drops of saturated ethanolic solution of cuproin. Purple to pink color is observed. With benzoin enzyme, sample solution should be weakly acidic and it is placed on filter paper along with a drop of 5% alcoholic solution of benzoin enzyme and then ammonia. Greenish coloration is observed. Test with ammonium thiocyanate and O-toluidine or paraphenylene diamine. Reagent solution is prepared by adding 0.1 gram of O-toluidine, 0.5 gram of ammonium thiocyanate and 5 ml of acetone. This is also a filter paper test. So a drop of reagent solution is placed on filter paper followed by a drop of slightly acidic sample solution. A light or dark blue stain is observed. Test for manganese. It is tested with sodium bismuthate to form purple color. Test with ammonium peroxydisulfate and test with sodium phosphate which forms pink precipitate. For the testing of lead, dithezone solution is used, that is prepared by adding 2 to 5 milligram of dithezone in 100 ml chloroform to 1 ml of extract. Extract would be neutral or slightly alkaline. 2 drops of dithezone solution are added. It is shaken for 30 seconds and the green color of the reagent will change to red. Burtonian lines or lead lines are blue lines on the gum surface caused by chronic lead poisoning. Lead diacetate is also known as sugar of lead. Test for zinc. Uh, Test with hydrogen sulfide in which white precipitate is formed. Test with potassium ferrocyanide in which blue-green precipitate is formed. Test with ammonium mercuric thiocyanate or copper sulfate test in which violet precipitate is formed. Confirmatory test techniques: UV visible spectroscopy, AAS, ICP-MS, and BAL or British anti-leucide dimercaprol 2,3-dimercaptopropanol is a chelating agent used in the treatment of poisoning by heavy metals. It is used as antidote in metal poisoning cases. Thank you for watching. For more such videos, like, share and subscribe my YouTube channel Vidhi Vigyan. Thank you.